All right, time to go teach. Hello and welcome to this episode of Tutors, where we are looking at enzymes. Enzymes are a specific type of protein that helps us perform specific chemical reactions. So we're going to look at them and how they work. Targets of the day, there's a few. First, define what an enzyme actually is. That's our, beginning. Next, explain how an enzyme is going to function. Because it's not enough to just know that enzymes exist and what they do. Now we have to understand how they do what they do. Then we're going to look at labeling and describing the different structure that makes up an enzyme. What different parts does it have? And finally, we're going to go into comparing and contrasting something called inhibition, ways to turn off an enzyme. So we're going to look at a little bit about enzyme rates, what they do with that. So, first things first, vocab, enzyme. It's a biological catalyst. But what's a catalyst? Well, a catalyst is a substance that speeds up a chemical reaction. But it's really important because a catalyst is not consumed by the chemical reaction. In some ways, it doesn't take part in the chemical reaction. So you start off with your reactants, and if you have a chemical reaction, you end up with your products. Well, if a catalyst is used, the catalyst enters the chemical reaction, and it also exits the chemical reaction unchanged. So it can go back to the beginning and interact with the reactants again, helping them become products. And then it can do that again and again and again and again and again. So an enzyme cannot be consumed by the chemical reaction. The actual chemical reaction cannot have a permanent effect upon the enzyme. If it does, it wouldn't be a catalyst. It would end up being reactant and product. So how does an enzyme actually work? To understand this, we're going to use a little graph. We have energy levels, and we have, on the bottom, we have our reaction. So the beginning of our reaction is over here, and the end of our reaction is on the far side. If we were to look at a simple chart, the reactants would start with a certain amount of energy. And in this case, this reaction releases energy, so the product's energy level is lower. That means that energy has been released. But even though energy is being released, you have to put some energy into the system to actually cause the reactants to become the products. They don't just naturally become the products for no reason. There needs to be sort of like a little push. Something needs to happen to give the reactants a little bit of energy so that way they can fully become the product. That difference from our reactants to the amount of energy that they require to become the products is called the activation energy, or the energy of activation. If the activation energy is not supplied, the reaction doesn't happen. So it's really important for our reactions to take place for them to be supplied a proper amount of activation energy. If they don't get that, we don't end up with our product, and that's a really bad thing because we have a lot of chemical reactions taking place within us, and we need them to happen. Otherwise, we wouldn't have energy after eating. There would be no point in eating. We'd never get the energy from it. So enzymes are going to be involved, and what they do is they lower the amount of activation energy that is required. And because they lower the amount of energy of activation that's required, that causes the reaction to happen at a faster rate, and so we end up being able to perform those different reactions within us. If the reaction still took that extra bit of energy, it would happen at a slower rate, and that wouldn't be beneficial to us. So enzymes speed up the chemical reactions by lowering the energy of activation. They make it easier for the reactants to become the product. That's what enzymes do. They allow for the transition of going from reactant to product to be easier, to cost less energy, and thus they end up speeding up our chemical reactions. So how does this all work out? Well, first off, we have our reactant, which we will be calling the substrate now. The substrate is the reactant. They are an interchangeable term. So we have our substrate and we have our enzyme. Now the substrate and the enzyme are obviously going to have to interact. To show this, we're going to look at a certain structure that's on the inside. So if we take a cross section of our enzyme and we look, this shape on the inside is called the active site. And it's pretty clear the substrate and the active site look like they would fit together. And they do. The substrate and the active site are going to be very, very closely related. It's almost like fitting your hand into a glove. The substrate has to be able to fit into the active site. This is very specific to enzyme and substrate. Specific enzymes only work for specific substrates. Different substrates are obviously going to have different shapes, which means different enzymes are going to have different active site shapes to make sure that they can accommodate a different substrate. In this case, our substrate is able to fit right into our active site. And when it does, we form the enzyme substrate complex. The reason that the enzyme substrate complex works is because both of structure, the shape of these two molecules, and also due to polarity. The polarity of these molecules is going to line up in such a way that they're going to be attracted to each other a little bit. And that attraction allows them to fit together. Now, our water molecule can actually enter in, and we are going to have the reaction of hydrolysis take place. Once hydrolysis takes place, we have our enzyme product complex. We no longer have the substrate inside of our enzyme. We now have just the product. And then the product is going to be released by the enzyme. It goes off into solution. The enzyme has not been changed. It has not been altered. And now its active site is open again. So another substrate can enter into that active site. And the reaction of hydrolysis can take place again, splitting our two different monomers apart from each other, and repeat, repeat, repeat. So that's how this enzyme specifically works. And just so you're aware, that's how hydrolysis takes place. It's done using an enzyme, and that's also how dehydration synthesis takes place. That's why animals build glycogen and plants build starch. We have different enzymes, and even though we're going to have both alpha glucose that we're putting together, the reason that we put our alpha glucose together in a different way is because we have different enzymes in us than plants. It's all because we have a different enzyme that we build a different type of molecule.